introduce in this symposium. Dr. Matheus Freitas Fernandes-Pedrosa holds a degree in pharmacy from UFRN and a PhD in biotechnology from University of São Paulo. He was a senior researcher at Instituto Butantan and is currently a full professor in the Department of Pharmacy at Federal University of Rio Grande do Norte, as well as a CNPq Productivity Fellow 1B. He has extensive experience in biotechnology, focusing on scorpion peptides, antimicrobial peptides, snake proteins, and immunobiological production, holding 6 patents granted by INPI. He leads a team of 17 researchers and has supervised over 80 research projects among PhD, masters, and undergraduate. Dr. Fernandes-Pedrosa has international collaborations with institutions such as the University of Rouen, France, University of Granada and of Seville in Spain, and Nagasaki University in Japan. He was the executive manager of the BioInnova at UFRN Incubator and is a member of various ethics committees and councils, as well as leading projects funded by CNPq, CAPES, and FINEP. So, Professor Matheus, it's a pleasure to have you here.
Thank you very much, Professor. Do you have the pass? Okay. Good morning for everyone. Let me see. Okay for me. Okay, good morning, everyone. I'm Matheus Pedrosa from Federal University of Rio Grande do Norte, Brazil. I will show here a little bit that we have studied in the last 16 years. The Norte is from Brazil. We have located at the Norte State. The Norte State is the state of Brazil, closer than we have here studied about the teachers stigmurin scholars that is predominant. Norte is from Brazil. Teachers stigmurin is morphologically characterized by yellow scorpions from about 7 centimeters. These yellow scorpions presents usually a black strip on his back. Unfortunately, I cannot point to on its back you can see a black strip. Its head, a black triangle. The main peptides of the chemical composition are represented by neurotoxins, antimicrobials, We have performed the transcriptomic analysis about 14 years ago in the Amsterdam study group, but it's a very important study because following the transcriptomic results, we have identified the high abundance transcripts present in the Venom gland of Tithys stigmuds. We can observe that about 70% of the molecules represented other possible venom molecules and toxins, no toxins molecules. And we can highlight an anionic peptide, AMP, alpha and beta KTX, metalloprotease and hypotensin as the main molecules expressed in this specialized gland venom of Titus stigmus scorpion. So after this step of transcriptomic analysis, we have identified and isolated the peptide that we have named stigmurin. Stigmurin is a peptide present in 17 amino acids in his primary structure, about 1.8 kilodaltons. At the first moment, stigmurin was positive against gram-positive bacteria and yeasts of genus Candida, and stigmurin was not effective against gram-negative bacteria. In the biocompatibility assays, we have observed that stigmurin was no toxic for hemolysis experiment, and for normal cells, viral E6, stigmurin was no toxic until 17 micromolar, and tests against cells SIHA for adenocarcinoma of women, stigmurin was toxic until 70 or more, 70 micromolar. So in the next step, we have used the primary sequence of stigmurin as reference and based it on literature to bioinformatic analysis that we have observed in specific points on its structure, on its primary structure. We have inserted lysine and focus on to obtain new analog peptides with increasement of their alpha helix and the positive charge to better interaction with the membrane of bacteria and to reduce the cytotoxicity. So we have obtained 31 analog peptides. We have deposited this patent in 2015. And now, last year, we received the patent letter in 2023 from ENPI from Brazil. Here we have this. Ok. Aperta e segura. Ok. Obrigado. Thank you. No, with me. Okay. Here we have presented the primary structure of stigmurin, amidate sequence. We have the 8 analogs that we have studied in more details. We can see some new peptides. We have inserted lysine and in 2 positions, replaced by serine or glycine, for example. In 3 positions, 7 and 14. In 4 positions, for example, in positions 1, 2, 3, and 4. And the most aggressive mutation we have obtained Stig 31, 5 replacements for lysine. We can observe for bioinformatics the increase of alpha helix, the increasement of positive charge. So we have on the next step, we have analyzed the native peptides at the first time in comparison with the analog Stig A15 here by experimental NMR 800 megahertz in Rio de Janeiro in collaboration with Professor Fábio Almeida at Federal University of Rio de Janeiro. The final result of the structure, Professor Jarbas was very important in the structural resolution of these both molecular structures, and we can observe the stigmurin, the start of the alpha helix in position serine 7, until phenylalanine 16, while if in the comparison we can observe the start of alpha helix in analog peptides 2 amino acids before isoleucine 5 until phenylalanine 16. Here we have confirmed the alpha helix predomination and that alpha helix was increasement in the analog peptides that we have generated in our group. Following with the pharmacological test with stigmurin, the native peptide, we have performed we have performed experiments focused on one healing activity of this native peptide. So we have performed these experiments in mice. We have promoted the wound in the skin of the animals. In first column, the uninfected control. In the second column, we have promoted the wound and infected with Staphylococcus aureus. In the third column, we treated the infected wound with Staphylococcus aureus with stigmurin 1 microgram per microliters. And we have followed the the activity for 7 days with one analysis per day. So we have performed the counting of the colonies. We can observe the decreasement of the counting of the colonies. Here we have the stigmurin, and in comparison, always with vancomycin, the market antibiotic for this purpose. So, in the next step, we have performed some more details in experiments focused on 2 analog peptides, named STIG8 and STIG18. Here we have the new sequence based on stigmurin. Stigmurin, the first sequence, the 18 two mutations, serine 7 and glycine for lysine 10. And 18, we have 3 mutations in positions 7, 10, and 14. We have analyzed the increasement of hydrophobic moment, the decreasement of hydrophobicity, the increasement of cation charge, and here we have presented the molecular mass of each peptide. We have analyzed the stability of these peptides by circular decrease in the first moment, and we have analyzed both peptides in different media, water, PBS, SDS, and a range of TFA, beginning at 20% until 7%, and we can observe in these results that both peptides show the predominance of alpha helix above 50%, and stability in different media, in special TFA and SDS. Experiments focus on variation of pH in a range of 3 to 9. Both peptides showed a high stability, in special STIG18, with maintenance of their conformation, structural conformation, about more than 50% of helix. And the third and last experiment, we have rating the peptides until 98 degrees. After this, we have cooling both peptides, and we could observe the discrete light difference of conformation when we reach until 98, but the conformation returns to original structure in the end of the reaction after cooling. So these experiments taken together, we can observe an important stability of these peptides in different media, pH, and temperature. So in the next step, with both analog peptides, involving both peptides, we have analyzed by dynamic circular, molecular dynamic experiments by bioinformatic analysis, the membrane peptide interaction. And we can observe for the first column, STIG8, second column, STIG18, here. At the first moment, in the experiments involving both peptides and membrane of Staphylococcus aureus, we can analyze that the peptide binding with the membrane by electrostatic interaction at the first moment, and we could observe also that the first contact occurs in in basic and polar regions of the peptides, basic and polar regions. And the third moment, the peptides inside deep the membrane involved with the phospholipid group to promote the lysis of the membrane of the bacteria. Following for the next experiments involving biocompatibility tests of both analog peptides, we have analyzed by the cytotoxic in tests with hemolysis and larvae of the species Galera melonella. In both experiments, in all concentrations that we have tested, the peptides showed that was no toxic against this model of cytotoxic experiments. And for in table 3, we have performed and showed here the determination of MIC and MBC of stigmurin in both peptides against gram-positive and gram-negative bacteria. So we can observe from against the gram-positive bacteria, stigmurin was effective according to the preliminary experiments that we have shown in the beginning of the presentation. In comparison with the analog peptides were more effective in lower concentration, was effective from gram-positive bacteria. And from negative bacteria, gram-negative bacteria, stigmurin was confirmed that was not effective, while both analog peptides were effective against E. coli and Pseudomonas aeruginosa. In experiments conducted by focus on treatment of early biofilm and late biofilm involving both peptides on biofilm of multi-drug resistant Staphylococcus aureus clinical isolates. We have performed in these experiments involving 7 streams of Staphylococcus aureus, and we have tested in 3 concentrations of both analog peptides and 3 concentrations of vancomycin, our positive control. We can observe for the majority of the streams that we have tested, we can observe important values of inhibition of early biofilm. The first side is early biofilm, and on the left, on the right, we can observe the late biofilm. So we can observe values important as 78%, about 60%, 57%. So we can summary in the result between 50 and 75, 78%. It's an important value if we compare similar values found with vancomycin. For late biofilm, we have important inhibition too, but the values was not so higher than early biofilm. We can observe the variation between 35 to 60% the better results, and these results follow the results presented by vancomycin. So we have the topography of electronic scanner, electronic microscopy. We have observed colonies without treatment and colonies treated with Stig, in this case for early biofilm, treated with Stig A and Stig 18. We can observe the decreasement of the colonies, the fragmentation of the colonies, the pore formations between the membrane of the colonies. So this picture is about the late biofilm for the Staphylococcus aureus, the same streams, and we can observe the more aggressive result in stage 18. Regarding stage 18, we have 3 mutations. The result is more aggressive if compared with stage 8 with 2 mutations, 3 insertions of lysine. So to conclude the pharmacological tests focus on both analog peptides, we have infected larvae of species Galera melonella with multi-drug resistant Staphylococcus aureus, clinical isolate. And we have analyzed the survival of the animals for 7 days in comparison with infected control, vancomycin in higher concentration, STIG8 with 2 mutations and 2 concentration, STIG18 in 3 concentration. We can observe that vancomycin was able to maintain the survival of the animals for 7 days, and STIG8 and STIG18 for 5 to 6 days, and a better result for the STIG18 in the higher concentration. So we have 3 possibilities of 3 hypotheses of mechanism of action of our peptides. We have the possibility of toroidal pore, we have the possibility of the carpet and barrier stave. Our hypothesis, we don't have results enough to conclude to close a hypothesis, but our hypothesis, based on our preliminary studies, is the mechanism of action is focused on carpet, when the peptides in agglomeration in peptide forms an overlay of the peptides that can disrupt the membrane, acting as similar as an emulsifier agent to promote the disruption of membrane after the overlay of the agglomeration of the peptides. The dynamic simulation I did not show, but the dynamic simulation was performed with 1 peptide and with the agglomeration of peptides. And this point is important to reinforce the possibility of the hypothesis of carpet model. Other important point that we have continued this study, maybe on NMR in solid phase, is focused on the target, molecular target that our antimicrobial peptides can interact in the positive or gram-negative bacteria. We can highlight teichoic acid as a possibility and lipoteichoic acid as a possibility too for gram-positive negative bacteria in the membrane interaction of the peptides, and LPS for gram-negative bacteria. These possibilities that we have focused. We had continue these experiments to obtain results in more detail with the interaction with collaborators in Europe. So now we have, in parallel, we have performed the second patent focused on new analog peptides based on native peptides as a reference. And we have invested efforts to study in more detail the native peptide TSP2. TSP2 is a second majority peptide present in Titus stigmurin scorpion, belongs to the class of AMP. The stigmurin was the majorities, TSP2 the second majority. And for this case, we have analyzed the primary structure of TSP2, and we have inserted lysine or arginine, or both lysine and arginine. We have obtained 41 analog peptides. We have performed a study of top-down. We have generated more than 100 peptides, and for bioinformatic analysis in more details, we was closing the sequence, and for top-down analysis, we have closed in 41 peptides. Other peptides that we have preliminary studies in our group is the majority class of peptides we have observed in transcriptomic analysis, the anionic peptides that we have named in our first article with these peptides, STAMP, anionic peptides of Titus stigmuris. This peptide is chelating for ions copper in a proportion 1 peptide for 5 molecules of ion copper. It's a big sequence, about 44 amino acids. And we can observe the predominance of aspartic acid. We have identified this peptide in mass spectrometry, and we have evaluated the chelating properties, the chelating properties with other ions like iron, like magnesium, calcium. But the most promising ion was the result focused on ions copper. We have analyzed the immunomodulatory activity in macrophage cells and have performed other experiments focused on this peptide. And we intend to obtain a product in the future to chelating ions and deposit a new patent. Another peptide that we have preliminary studies in our group is a high-potency peptide. High-potency peptide presents a primary structure about 25 amino acids with the domain characterized for high-potency, the domain PPA. We have performed in collaboration with Professor Valdir Braga. The experiments focus on vasorelaxante activity. We have studied also the Bragg-Senin potentiation activity. We have tested the efficacy of these peptides in some antimicrobial models. We have analyzed the toxicity of these peptides. So we have to continue this study to study in more details, but we have promoted some alteration, some chemical alteration in this on X structure. We have deposited a patent focused on a new candidate for antihypertensive drug in pharmaceutical area. So our perspectives, we have 7 native peptides, we have 2 analog peptides. We have continuous in collaboration with Professor Sylvie from University of Rouen in France. We have focused on mechanism of action, focus in the first moment with Pseudomonas aeruginosa. We have already studied this focus in France, but we have continued for the group SCAPE, Enterococcus, Staphylococcus, Klebsiella, Cinobacter, and Enterobacter group. The group SCAPE is the group very important for OMS because the high resistance of these microorganisms. We have to continue the NMR analysis from the other native and analog peptides. We have to continue topical formulations, focus on the studies to apply this product as wound healing treatment. We have now performing results that not published yet for focus on synergism. We have observed in for some antibiotics, synergism or additive effects. The anti-insect activity we have also started nowadays. And we hope that in the future, nowadays, if what's possible, but in the near future, a partnership with our pharmaceutical industry. We have a patent letter, but we don't have now a pharmaceutical industry to make business, to invest in our molecules. So we wait for opportunities to deal with pharmaceutical industry. To conclude, some important collaborators of our experiments, Professor Fábio Almeida from Federal of Rio de Janeiro. We always, every year, we have to stay there for two or 3 weeks, sometimes 4 weeks. We have usually traveled 2 or 3 times in a year for Rio de Janeiro. And this is very important because we have the equipment with 800 megahertz to analyze our molecules. We have also collaboration with Professor Jarbas Rezende that is here in our Congress. Jarbas is very important to synthesize our peptides and resolution of the structural conformation of our peptides that we have acquired the date from NMR 8 megahertz. Professor Renata Mendonça is so important, our collaborator from Chemical Institute of Rio Grande do Norte. This picture was in Department of Chemical from Federal University of Minas Gerais, here in Rio de Janeiro. And we have also to thank Professor Rafael Ximenes from Federal do Rio de Janeiro for the Staphylococcus aureus multi-resistant clinical isolate, and Professor Sylvie Chevalier from Université de Rouen in France. We have started the experiments focused on mechanism of action of our peptides, not only 2 analog peptides, but a pool of analog peptides against Pseudomonas aeruginosa. And nowadays, we have 2 postdocs in performing experiments in Universidade de Granada in Spain and Universidade de Seville in the same country. They are performing the production formulation tablets for antihypotensin toxins, and dressing for that wound treatment with our peptides is a project that we got the success in PQ last year. The postdocs will stay 6 months in Seville and Granada. After this, they will to France for more 6 months. Okay, I'd like to thank all the collaborators. We have collaborators here at Universidade from Brasília, Professor Elizabeth Ferrone, Professor Werner Triptel. We have now got the 2nd project approved in coordination by Professor Werner Triptel. Part of our group, postdocs, doctoral students, master, and scientific initiative students, and UFRN Natal. Thanks for your attention.
Thank you for your presentation. And now it's open for questions.
Congratulations with all the results and the beautiful presentation. I was interested in particular in the anionic peptide, since it can chelate copper. Do you see any application in clinical toxicology in terms of metal intoxications to treat patients, or do you expect that the side effects would be bigger because it may also chelate calcium, and then of course it is very dangerous for the heart? What's your opinion? Is this molecule a potential drug for an antidote?
Yes, this is a very good point. We have tested, we have only performed preliminary tests in culture cells, and we have only proved that this peptide was not toxic for fibroblasts and other kidney cells, only for this, but for iron calcium, the peptide was not efficient. The better efficient, the stronger binding interaction occurs with iron copper only. Iron, calcium, magnesium, it's not so good, the chelating. So we plan to use these peptides maybe on agricultural area for promoting new molecules for anti-insect activity, maybe a possibility. And the other possibility is for cancer cells in the cerebral, brain cancer cells, or some diseases in brain. But we have to study deeper to apply these peptides. We have only preliminary studies. Maybe we have in our hands important molecule. We have to study in more details to apply on toxicology for pharmacological approach. Okay, thank you very much.
Does it have causative? Yes, isolate with se já vem com íon quando você purifica, ela vem sem.
This is a good point. It's not constitutive. To see, we could observe the interaction in mass spectrometry, but we have to put in contact in experimental analysis peptide with ion and observe in mass spectrometry. but it's not constitutive. It's very good. It's a very good point. Thank you.
Very nice talk. It's very nice to see you again after so many years. So I was interested in stigmurin. So I saw that you just understand a helix by using NMR. And my question is, what kind of conditions did you use to see these NMR structures? Because what I'm seeing is that I'm seeing a coil that was transforming in a helix when I have the peptide binding in the membrane. And I saw that you basically see a helix. So what kind of conditions did you use to elucidate the structure?
Thank you, Professor Otávio. It's a very good point. We have used the condition of TFE about 40–50%. We have now obtained the new condition involving SDS, 20 millimolar. We have other conditions that we have planning to study for a new paper, but this point was very discussed in our published article because almost all of the supervisors focus on this point, but we have at the first moment in the first paper we have performed these experiments involving a range of TFA. Because of this, in the first experiment focused on NMR, we have selected TFA in two concentrations, 40 to 50. But we know that in the second moment we have to present in other condition in special SDS. A lot of supervisors of the paper required SDS. But it's not easy because it's not easy to use the equipment, to use the resolution. We have support of Professor Jarbas, Professor Fabio, and nowadays we have to conclude the experiment focus on SDS to present for the literature. It's a very, very important point. Okay, thank you very much. Okay. Thank you, Professor. Okay, now I will present the next researcher, Professor Octavio Luiz Franco. Octavio Luiz Franco has a degree in biological science from Federal University of Ceará and PhD in biological science from the University of Brasília. We carried out his first postdoctoral research at Embrapa, following another in the University of Edinburgh, Scotland. He carried out work missions in several countries, including Australia, Israel, Portugal, Cuba, Netherlands, among others. Completed his first sabbatical of the University of Wisconsin, USA, and second in British Columbia, Canada, focused on infectious disease control. He currently works as a professor at the Catholic University of Brasília and the Dom Bosco Catholic University. Additionally, it coordinates the National Institute of Science and Technology for Bioinspired Products. Dr. Franco holds the position of Research 1A and works as consultant of National Council on Scientific and Technological Developments in PQ. Furthermore, he is a member of Brazilian Society of Biochemistry and Molecular Biology and American Society of Biochemistry and Molecular Biology and the American Society of Advances of Science. His research focuses on development of bioproducts and bioprocess with the emphasis on the control of pathogenic microorganisms. Dr. Franco has authored nearly 500 articles, mentored hundreds of students, and holds dozens of patents for products that have entered the cosmetic and animal health markets. Dr. Otávio, thank you very much.
Thank you, Matheus. So thank you once more, Mariana, to invite me. I'm always glad to be invited by Mariana. It's always a pleasure to be here. I was a student from here. Mariana always remember me, so I'm very happy. And Wagner also. Where is Wagner? Oh, here he is. So thanks a lot. For today, I will talk a little bit different of the other talks that have been done in the last months, because Mariana just asked me to focus in mastoparans. I'm not seeing me like a guy that is studying toxicology or something. I'm seeing me like a Lego builder. So I just constructing peptides, and when Mariana gave me this invitation and talk, oh, why you don't talk about the wasp peptides and other things? I was just thinking, okay. And then I discovered that we have done a lot in the mastoparans. And I will start my talk talking that for the beginning, I don't want to work with mastoparans. I remember that I have one student, a PhD student, a very good one, that the name is Osmar, and he just asked me to work with mastoparans. I say, no way, I don't want to work with venoms and toxins, because probably we'll never sell that. I'm always trying to construct a product for animal health, or human health, or for cosmetics or something. And in my mind, I was just thinking, could be very hard to sell a cream with a toxin of a snake or something. But then this was not true. I will show you that basically the people do not make this connection like a marketing place. And I remember that this guy, I say, no, we don't want to work with these mastoparans. And then he lies to me. He say, okay, Octavio, no problem. And then he start to work in my lab with the mastoparans. And since I have a big lab, sometimes I lost the control of my students. And one day he just asked me and say, okay, Octavio, I need to show something. And I say, okay. And then he showed me very, very good peptides. And I see the sequences, and I do not found the sequences in my mind. So I do not remember this kind of sequence. And then he told me, okay, these are toxins from wasps. And I say, what? And the guy say, take it easy, calm down, don't be rage. I will show very nice results. And when he showed me all these nice results, I say, okay, Osmar, I love you more. And now I love you also the mastoparan. So it was interesting, because in the beginning I don't want, and now my 3 main products are coming from toxins. And this is, for me, is quite surprising. So today I'm just coordinating 3 institutes: the SC Nova Biotech in Campo Grande, that is involved in bio-inspired products for animal health, and now agriculture, because the government asked me to focus also in pathogens of plants. The Centro de Análises Proteômicas e Bioquímicas, that is my old center, now 25 years. It's a lot of time, I'm getting old. And the National Institute of Science and Technology for Bio-Inspired Products, that this year make 10 years. And today I will talk a little bit about the mastoparans. So I think that this is... Okay, normally we start with natural products. I always, like most of people here in Brazil, started purifying peptides. I never did this before with wasp venoms. And then we work with clavanins, with Centritis muricatus, that is CMP5, and the plant peptide. So we just find a lot. And then this lead me to a problem, or many problems. We can publish a lot with these peptides, but we never go to the market. It was also a big space between the product development and the paper. Why? Because we have a lot of complexity, low activity, high specific activity, aggregation, difficult to purifying, so many problems that now we develop some platforms to transform these peptides in something that could be useful for the industry. So mastoparans are targets for peptide design, so we can use these mastoparans as the base of new products. So a very nice charge distribution, a fragment excision, so we can cut this peptide and use part of them. I can apply D enantiomers in order to avoid the proteinase destruction. We can use alanine scans, and we can use deamidation to protect them, and then they have multiple targets. So we can also find some mastoporans that destroy cancer cells, that could be very to erythrocytes, to fungi, and also to kill gram-positive and gram-negative bacteria. So once we have been using these tools to construct new peptides, we started from the beginning. So my first mastoporan that we published was a derivative of the mastoporanel. So mastoporanel is a very short peptide, and then one thing that I like it most is to study the distribution of the peptides and the amino acids inside of the peptide. How I said, I think in the peptides like a Lego. So I'm just thinking in the amino acids like blocks. My father was the first distributor of the Lego in Brazil. So I was the kid that was just putting the Legos and the pieces here in Brazil and trying all of them. So when I was very young, I do not have other toys, so I have only Legos. And my father is the guy that was thinking, okay, if you have a Lego, you'll be smart. So I have Legos all the time. So in the beginning, it was very funny. In the middle, was not so funny because I want a video game. And maybe I spend a lot of time with Legos. And then when I look to a peptide, I'm looking now like a pieces, and the amino acids could be distributed. And I always thinking that was very interesting because I have 20 pieces, and I can construct many different things. So with a Lego, I can construct like a house, a boat, a Batman, or something. Here I can construct anti-cancers, antifungals, antibacterials, and it was very, very interesting to understand how could I improve the activity against bacteria. So one thing that we observed that we need to change the hydrophobic moment and also the cationic position. So one thing that we done in the beginning, I don't know how to, no, how could I put a, no. Arrasta para o lado? Arrasta para cima. Ah, cá está. Ah, cara. Você tem que fazer um negocinho assim. Vocês são chiques demais. So, we just added an isoleucine 5, and we switched it by alanine, and we added an arginine, arginine number 8, and for this thing, we just named it E5R8. So, I improved the charge in the surface and thinking that we can just also improve the attachment in the surface of the membrane. And this was true, so we ran some molecular modeling, observed that I5R8 was much more attached to the surface of the membrane. And we observed that the MICs was decreased. So we really just changing 2 small pieces, and then we reach to an improvement of the activity. And this was also observed in the time QSIs, in which we observed that I5R8 was more faster to kill. Another thing is that we observed that the peptide keeps the mechanism of action in depolarization of the membrane. So we just compared the melitine, and we observed that we observe a similar thing that Matheus just observed, that we have like a detergent effect in destroying the surface of the membrane and allowing the changing of cations inside. And here we observe the effect of the peptide in a microscope, electron microscopy, and for 2 different bacteria and for candida. So we basically observed that the peptides go to the surface of the membrane and cause a terrible disruption. So we can just go further. And in this case, we just change a little bit inside. Another strategy was to add a tag at the end of our peptide. So we just have these mastoporans, and then at this point, I was interested to help in the sepsis model. And then we were also interested to reduce the inflammation. So we just make a huge alignment of many and many other peptides. And then we discovered that we have a combination of 5 amino acids that we try to put at the end of the mastoporan to decrease the inflammation. So what we done? We observed that we have a conservation of this FLPAA in many immunomodulatory peptides, and then we add basically to the mastoporans. And then in the beginning, no, sorry. Como voltamos, nunca mais. Okay. We just, first we try with clavanins, that I do not show the data here, and nothing happens. And then we try with mastoporans, and then we observe that we can change the activity. And then at this point, I was working with Cesar, and he called me and say, why you don't put your tag also in brevinins and temporins and see what happens? And then we inserted these peptides in many, many different other peptides to see the effect. So we basically have a toxic mastoporanel, and then we add this tag, FLPAA, that is founded in the data bank, and then we created the mastmol. The mastmol was used many, many times in many, many articles, and this was the salvation of my student, because Osmar created this tag that works with the mastoporan. So we make a lot of assays, and we observe that this mastoporan MO was very, very effective against gram-positive and gram-negative bacteria. And then we observed that in the time kill that it was very, very efficient by fluorescence assays. And then we just run a lot of immune response tests, and we observed that this peptide, when received this tag, decreased the interleucin 6, the TNF-alpha, and reduced the inflammation associated with the antimicrobial activity. So at this point, we discovered, okay, we can add an extra block. And when we add this extra block, we have extra function. So that's cool, because now I know that a multifunctional peptide like mastoporan could be modulated, and I could add some extra activities. And then we just make a permutation. What's a permutation? My question was, if you have the FULP, this tag, in a certain sequence, and if I change the position, if I put the pieces in different places and combine with the different peptides again, so we perform these permutations with all of them. At the end, we have 500 different peptides, and we tested all of them and discovered that at the end, we need the sequence in the right place. Sometimes I can change one on another, but it was not like a question of, oh, very hydrophobic position. We need really the phenylalanine, the isoleucines, and teprolines in the right place to obtain the immune response effect. And then we run a lot of animal tests and observe that we can control the sepsis until certain point. So we can control until 80% with a 16 micromolar of activity. And then we tested several of them, the toppest one, and once more, the mastoporan MO was the best one. It was incredible, because my student created from the beginning, we make a lot of combinations, we just change the tag for other ones, and nothing was so good like the mastoporan MO. So he was forgiven. I like him very much. He expended a lot of money of the laboratory, but it was a good, very good paper. And then we ran a lot of NMR structures in order to understand how this tag changing the structure of the mastoporans. And you observed that when you have this peptide in the mastoporans, we have a different combination that calls a kink. And this kink, it seems that binding to something that I don't know what is it, and then perform the immune response activity. And then, okay, we just make this huge experiment with a lot of peptides, and I was just thinking, okay, we can create more. So basically, we just put a tag, we just change one on another, and then we created a rumble, a mixture of peptides, 500. But maybe I could be more smart. Maybe I can use some algorithm to improve my hits, because it's quite expensive to expand 500 peptides to hit 1 or 2 or 3. Maybe I can use some strategy. So we use this Joker algorithm, in order to find in all mastoparans and other classes of antimicrobial peptides some identity components. Oh, let's see. I'm so terrible with this. So with identity components, so there are some amino acids that normally are found in these antimicrobial mastoparans, and there are some ambiguous components that change the classes, for example, a lysine by an arginine, or a phenylalanine by a tryptophan or something, and then are the wild cards. So the wild cards was used to create something that the nature did not offer me until now. So I add a piece over there that I cannot find in any place, and then we just add these fragments to scaffolds, to structural scaffolds that we can make more easy for the industry. For example, in this case, I was interested to helix. Most of people are interested in helix, including David Baker, that's creating the AlphaFold, because they are easy to predict. They normally are not very, very much aggregative. So quite nice. And then we just make a slide window. And then we create a lot of peptides. In this case, we created in the beginning almost 300, and then almost 120 different mastoparans. And I will not show all the mastoparans, but we have the R1 and R4 that was very, very potent. So was in the beginning, I changed one on another. Then I add a tag. Now we must use the strategy to improve the hits. And then we created these two different peptides. In this case, I have the same of you in TFA. We observed a helix, but when you add this in water, nothing happens. I have a lot of difficulty to see the NMR because there is a coil and you cannot predict the coil. It's like a crazy peptide. And so we have a very potent peptide, but the next question is, if I put salt or if I have temperature, do I have any problem? Because this is one of the limitations of the industry. See, if I have salt and block it, the positive and the cationic amino acids, so I have a problem because in plasma I have some salt. Normally in the creams and other things I have salt also. So we just tested against multiple concentrations and multiple in different salts, and we observed that these peptides do not have the salt not have any effect over these peptides. The same was in the temperature. We can keep this peptide active until 50 degrees Celsius, what is quite okay to go to the field. And then we just run some SPRs in Brisbane with Angeline, that is a suite of person, and then we observed that this peptides was very, very affinitive with the membrane. So we just created something, in this case, no, in the, oh, okay. In C, we have mastoparanel, and the R1 and R4, we have two different peptides generated from the mastoparanel that show more affinity to the lipids. What leads us to better understand the mechanism of action. So once more, we just run some molecular modeling experiments, and now we are just trying to simulate the peptides with the membranes, and also a different distance, because when I give the peptide was not just inserted in the membrane. We are trying to simulate what occurs when I have the peptide floating and then flow to the surface of the membrane and go inside. And what we observed, when you have these modifications, we have a more fast affinity of the peptide in the surface and also the movimentation inserted inside of the membrane. Now, one of the problems that we need to solve is because we need more computational power to have thousands of peptides over a huge membrane and see what happens, because it's always difficult for me to believe in that one peptide will do a lot of destruction in the membrane. Well, we tested other activities of mastoparans. So we tested also antiviral activities. We see that part of the mastoparans included the I5R8 and mastoparan modified the mastemol, impede the adsorption and penetration of herpes virus. We tested also in this big project with Caesar in the coronavirus. So we just tested, and here when you have this not so blue circles, it show me that the peptides avoid the destruction of cells, of liver cells, and the mastoparan MO protected almost all of them in different concentrations. Here we tested a lot of other peptides that are not so effective. In the case of mastoparan L, we don't see this protection, but when you see the MAST-MO, we can see the protection against virus. And then we just start this activity in order to try to understand how this peptide becomes also antiviral. It seems that this peptide bindings to the lipids that comes from the vesicles that the virus carried, but we do not find any evidence that this peptide binding directly to the virus, maybe to the membrane, but not to the virus. And then we have a lot of studies with anti-cancer tests, and then we show that MAST-MO, MAST-L, and other derivatives were capable to control the tumors. In here, we just combined it with gemcitabine and mastoparan, showing that we basically reduce the tumors a lot. So then we decided to go to a place that I could generate a product. So in the case of cancer, it's almost impossible, even because now I'm inserted in the pharmaceutical industry. I'm just coordinating the development strategy of MS in Brazil today, but they are not interested in cancer or infections. Basically, they are interested in other kind of like immune responsives and other things. So I was just thinking, okay, I have a lot of peptides, and normally they are much more interesting to peptides that they develop by themselves, not by buying for anyone from the university. And then I just focus in animal health. So I'm thinking, okay, I can contribute by improving the quality of the foods in our country. So if I have some peptides that decrease the bacterial activity, and then we do not have any residue, we just can use peptides with a green technology that avoid the contamination of milk, meat, and other things. So we just use the mastoparan-like peptides that we already created to test it against the Staphylococcus aureus that was very, very terrible for bovine mastitis. So in this case, we tested once more the MO, the R1, and the I5R8. So we just observed that these peptides were very, very effective against the Staphylococcus aureus that comes from the milk farms. And then we just collected Staphylococcus in many parts of the country, in Minas Gerais, in Mato Grosso do Sul, and in other, because we need to challenge our peptides with the worst that we have for the producers. And then we have some candidates. And then we just observed this effect once more, the helix and blah, blah, blah. And here are the three worst. The Staphylococcus aurora that destroyed part of the milk was basically destroyed by I5R8. Mastoparan MO is more or less, and R1 was not so good. So in this case, we just focus on I5R8, and then we observe that it's not effective against cow cells, and then Aurora versus the I5R8, we have an effect in 16 minutes. So in 1 hour, we just make an injections in the uber of the cow, and then we can see that. And then we observe that this peptide could be stable in milk. So one thing that is very, very important is if I use the peptide, it not be destroyed in the place that I'm using. So we just put in the milk. So we have 100% of the peptides with 0 hours, and after 6 hours, we have 20%. After 12 hours, we don't have any residue. So we have the effect of this peptide for 6, maybe 10 hours, and then we cannot find this peptide anymore inside of the milk. What reduce the levels of the toxins and the residues inside of the milk. So in this case, the industry was very, very happy because, okay, it is very, very effective, and then I do not have any residue. And then we just run a lot of fluorescence microscopy and observed by living dead cells that at 8 micromolar we can destroy, including the biofilms of the Staphylococcus. And then this is the Staphylococcus aurora. This is very terrible. When I talk about the producers with this stuff, they say, oh God, and then they give me this. And you observe that they agglutinated all the time. They are a little bit different of other Staphylococcus aureus. And here is atomic force microscopy. When we observe that in the left side of you, we have the staph, and when we treat it with the peptide, we have the dissolution and dissolve of the bacterium. So we just observed that the peptide also decrease the size of the staph. When you add it, we have a destruction and a reduction of the staph size. And you observe that at the same time, when you have some candidas that we remove it from the Uber, we have the same effect. And how it works? So we just run some experiments with cryo-EM, and observe that the surface of the peptide binding to the surface of the membrane, and then cause a curvature that makes something like this. So when you see this like a detergent, we observe that, oh, we can see the holes. But at the end, it's not a very, very well organized hole. It's basically like remove a vesicle in the surface of the bacteria and creating these vesicles and destroying step by step the bacteria. So now, how much time do I have? I finished it? 15? Ah, tá bom. Okay, sorry. No, because I was not thinking about the time, so if you give me more time, I can talk all the day about the peptides. So, but it will not take so long. So now we are just taking this strategy for Pantanal products, and we included, because in the beginning it was not included wasps, but now we started to sequence genomes of wasps from Pantanal also, to find a new sequence that I cannot imagine or figure out, because I always thinking, okay, I can construct some Legos with the peptides, but nature have much more knowledge than I am. So sometimes I need to look to something that I cannot even think about it. So one of the strategies is sequences a lot of animals in Pantanal. So we just started to sequence including wasps, to see if I have differences that I cannot manage by using my bioinformatic tools. So we are just sequences, and for now we have almost 2,000 bioinspired products that was also involved in tests. One of them that is derivated also for a toxin is a anti-mastitis, like you said before. It's better than the mastoparan I5R8. And then we just have this molecular dynamics and NMRs and other things. And we just discovered that we name it now MastPep, was very, very potent against a pool of Staphylococcus aureus again. So we just improved it a little bit more with the strategy of bioinformatics, something that starts from a mastoparan. And then we tested against many, many different things, and also against cows, because at the end I don't want to kill my cow, I want a happy cow. And then we tested macrophages, mammalian glandular epithelial cells, and erythrocytes, and nothing happens in this pharmacological window. And then we run first a model with mastitis in mice. So normally it takes a lot to have the cows, so the Uber of cows is huge. And then I was just thinking, okay, could be to see how the peptide go inserted in the milk or in the Uber of the mice, and see what happens. And what we observed is with 64 micromolar, we have a good activity against staph, and it works. So basically, we have a treatment with a MastPep, and it works. At this point, then I go to Ouro Fino Saúde Animal, and then they was interested in have something with us, because we have not only developed the peptide, we have understanding the mechanism of action, and now we have some in vivo activity. But then they asked me, okay, did you try with cows? And at this point, I do not have cows in my house. And then I always tell, we have a impasse. We have a difficulty between me and the company, and then they say, okay, if you try with the cows, we can think in a contract. And I say, okay, how much is the guy say X, would be a good investment, and then I buy 10 cows. So I buy 10 cows and put in a farm of a friend of mine that works, and make an agreement with my friend. I say, okay, you take care of my cows, Black, Tiger, Panther, and put names in all of them. So they are my friends now, very, very nice cows. And you can use the milk to produce cheese, and you pay for the meal of the cows, and I pay for the cows. And the guy say, may I do a barbecue? And I say, no, don't kill Black, Tiger, no. No, never. So we make an experiment, a controlled experiment first, injected the Staphylococcus aureus in the cows. So I have people in my team that are very, very good experts in animal tests, and then we used a very low number of animals, and then I observed a very huge standard deviation. So we can see a possible effect at 96 hours, and at 120 hours is quite okay after several treatments. It takes me a lot of peptides. I expend almost 700 milligrams of peptides to do these small tests, but this was enough. When I showed to the company, the guy say, okay, it seems that it works, but now we need to improve the number, and now in the next year you go to a field test with 500 cows. So they offer to us the possibility to pay the rest of the experiment. Another thing is that we observed the milk stability. So one thing that I'm just selling them is that I cannot be so potent like a ciprofloxacin or other antibiotics that they have in the market. But I never leave residue. So this could be important for exportation of the milk, yogurts, and other things. And I reduce the possibility of resistance, because if I don't have residue, the pressure of the selection is lower. And the guy say, oh, that's cool, this is very nice, could be very, very important for Europe. And then I think that's it. So after the treatment, after 6 hours, I cannot see any residue of the MASPEP, what is very interesting for us. So it's finished, not yet. We are just trying to sell our second product. The last one was a cosmetic, so that is not from our brand, and for this reason I do not talk about this. And it was very, very, a very good hype, because one of the persons that I was using our product, peptide base, was Katy Perry. So was very, very interesting, the effect it causes in the cell of the product. One day, Katy Perry just opened the purse in one talk show in the United States and say, oh, I like very much of this product that is for skincare. And then in the next day, they sell almost 600 flasks of the peptide. So it was something very amazing. So now I'm just thinking, if Katy Perry don't want to drink my milk with my peptide and say, okay, I love this milk, and then we can use more peptides to create some products. So now we are just thinking to improve a little bit more, because now is another hype is artificial intelligence. So what's artificial intelligence? It's just a different way to see the same that you are seeing. Now I'm a guy that is constructing Legos, but now I have a robot in my side that is telling me, don't do that. Okay, this you can do it. So I have an imaginary friend that probably I had when I have 10 years old that helped me to construct Legos. So so we created a platform to improve our peptides. So we're using a lot in order to avoid the errors and improve the hits. One important thing that I applied in this platform is because I always remember when my mother was hard with me. So when I do something wrong, I received a very hard penalty from my father and my mother. And I remember when my mother gave me a sandal in my bags and say, stop it, stop it. But I remember sometimes when my mother and my father say, good for you. So I apply the same in the platform. The negative one was much more powerful than the positive one. I give much more penalties when my platform commit a mistake than congratulations when the platform is improved. So we used Lime, that's a local interpretable model, a shape, and another IMP prediction algorithm that we developed with Caesar and other guys. And we add other kind of information. So at the beginning, I was just looking in pieces. So I was looking positive pieces, hydrophobic moment, distribution. But now I'm looking also to the target. So normally the peptides could do many different things. So it can create a hole in the membrane. It could bind in another thing like a protein or something. It could make a curvature in the membrane that could be negative and positive. So I give him to the system all this information. But because at the end, the artificial intelligence could provide me something good if I give a very, very huge data bank. So we spent last year just giving information about antimicrobial peptides, molecular mechanisms, the most important amino acids, and then we created this platform that we are testing now. And then we used these parameters like spontaneous curvatures, negative pressure, positive pressure, toroidal pores, the composition of bacterial cell, the capability to cell that, and then we associated one thing that could make more easy, that explainable artificial intelligence. So when you put all this information, he tell me, okay, don't put this amino acid in this point. Don't put this one in this point. For example, in this case, the blues are the negatives and the reds are the positives. My team like to be inverse. Normally for me it's more intuitive to red be negative, but not in this case. So for example, aspartic acid, never use it for antimicrobial peptides. If you use it, you have a very low probability to hit or to create a very, very potent peptide. Otherwise, when you put the lysine, we have much more chances to hit to the goal. But you can say that in some cases, in very small cases, the aspartic acid could be there. So I have very small spots in red that show me, okay, don't use it in 99.9% of times, but sometimes you can use it. The lysine is the same. I can use it in 99%, but 1%. And then comes our brain, because at this point, it's not just to give to artificial intelligence and then receive it something and say, oh, thanks, artificial intelligence. If it was so easy, we can cure everything, but it's not so easy. It's much more complicated than I think. And then we created these new peptides, and now we are using something that we know, we name it molecular reinforcement learning. So our prediction model, with all this information, create the peptides and classify the AMPs and non-AMPs. And then we have anti-new Klebsiella or something, and then we have a high reward sequences, and we have chemicals restrictions, and then we have a sequence generator that gives me the new sequence. And then I go to the bench and test and say, okay, now I have this, and these are okay, these others are terrible. And then we return the information and say, okay, thanks for this 4, I hate you for this 500, so we start again and start again. So we spend a lot of time just synthesizing, testing, and returning information, and synthesizing, testing, and returning information. For now, after 3 rounds of these strategies, we have 286 sequences. Among them, 4 are from mastoparans, and 1 is very, very similar to mastoparan MO. Once more, thanks, Osmar, for being a hard head for me. And we have now 15 new sequences that we are trying to prove that are very, very good. And this is the PepK10. It works very well. It's not like I like it, because I don't know if it's perfect. For now, nature is much better than me, but we are trying to improve it. So now we are very near to have a very, very good peptide. And then what's the new strategies for us? So first, better understand the molecular mechanism of actions of these antimicrobial peptides, because until now, I really believe that we don't know too much about the mechanism of actions. We know that destroy the membrane, we know, but we don't know almost nothing. every time when you challenge this bacteria with the peptides, for example, in this case, we discover so many things when you have a resistance of these peptides. We make a very nice paper with proteomics, and we elucidated at least 25 mechanisms of resistance with the peptide. So basically, the peptide is binding something and give me a lot of more information that I need to use it to create peptides that will avoid this resistance. So the new targets for me could be important. Another thing that we have discussing a lot with pharmaceutical companies is they don't like the idea to have a peptide that binds into a membrane. A membrane is not an easy target, so it's not a very, very replicable target. They want something very, very specific. For example, what lipids and what part of the lipids, the peptide binding, and how it works. So when I say, okay, I have this antimicrobial peptide, okay, but what is the target? And I say, could be membrane, could be a protein. And the guy say, okay, Octavio, very nice paper, I don't want it. So they are very restrictive with this. They like the idea to find something with different protein or something. And maybe it could be this. The new strategy could be the redesign the mastoporans to a certain different protein and then created something that could block it with a very high specificity. So I think that is it. I always trying to show like a history, is because science is always part of my life and peptides is part of my heart. So I like very much of this, and I like very much of my partners. I have a lot of partners in many parts of the world. And now we have a lot of funds because this is quite expensive, how you know very well. So we have CNPq, CAPS, Fundec, that's the foundation of Mato Grosso do Sul. We have the cosmetics, IBA, that is providing us a lot of funds to discover new creams. FAPDF, the MS, the pharmaceutical one, FINEP, OneSkin, that we sell the first product with them. Peptides, that is one of my startup companies, and the Instituto Vitanova, that is providing this immunomodulatory response. And these are, oh no, come on, my team. I need these guys. They are the responsible for everything. Yeah. All right, come, no worries. Because it's a lot of work here, and these are the guys. So in the left side are my team in Campo Grande, and the right side is the team here in Brasilia. Now it's a little bit higher. This year was very, very good. A lot of people just come. And I always thank you to these guys that are working every day in the lab and have all the frustrations to discover 499 peptides that do not work and one that is working very well. This is the Insta and the TikTok and the part of distribution of science. I do not make short dances. I do not do this, but I tell a lot about how to be a scientist, or how could be important to be in love for the thing that we are doing all the days. Okay? So thanks once more, Wagner, for all the partnership. You are always very, very important for us with the part of mass spectrometry, Mariana, about the discussions about antimicrobial peptides. Thanks a lot, guys.
Okay, thank you very much, Professor Otávio, for the great talk. It's open to questions.
Thank you, Otávio. Great talk, as usual. By the way, I also love playing with Legos.
I can imagine. When I think in one person that likes Legos more than me, I think that is you.
Yeah, I had a great time in Denmark. So, I was thinking about one of the peptides, the tagged one, that has antimicrobial activity, has anti-inflammatory activity, but has some influence in sepsis, but not that much. I would expect more for that peptide. So I was thinking, maybe this anti-inflammatory activity is interfering with the antimicrobial activity of the cells. Did you test something on phagocytes?
This is a very good question. In fact, when we just submitted for the first time, the referee just said that it was not a very good strategy, because when I reduce the inflammation, I probably reduce also the defense against the microorganisms. So I think that we did not test it, but I think that you are right. So we have this effect when you reduce the inflammation. Part of the animals basically die, not because we don't have a correct effect, but because he lost part of the defense, and then we do not have the protection that we expected. In fact, when I compare the tag with the peptides and the tag with the other peptides, I don't see an improvement very much with the peptides without a tag. So we have something like 10 or 12%. That was enough to convince the referees that it was a nice history, but was, for me, was not a really amazing improvement. I expected much more. Another thing that I do not show here is when I have only the tag, I probably need to have the effect. But the effect only the tag is very low. So I can see the pattern in the bioinformatic analysis. When I add to the peptides, I can see something improving, but alone is not working very well. So at the end, I see an effect, but I don't know nothing about the mechanism of action of the response, of the immune response. What I have been learning this year is that when you have something very difficult with microbiology, is much worse than immunology. So immunology is I know that part of the people here could be from microbiology, but the immunology is terrible. You know, so many ways, so many things that could happen. So and it's very unpredictable. But yes, the question is very good. It's a question. Maybe we can test the macrophages and the leukocytes and see the migration. I think that we never tested this before.
We're doing that, so if you'd like to.
Yeah, for sure. Good. Okay.
Jarbas, okay.
Oh, fantastic results, Otávio. I was just wondering about that peptide, amastoperam MO. Did you check something regarding the membrane insertion of the peptide N-terminus? Because probably that phenylalanine residue at the N-terminus has something to do with it. So perhaps checking some analogs and the effect of this phenylalanine on the membrane insertion could give some insight on the great results it has shown. Thank you.
Well, I never mark with the fluorescence this one to see if the insertion is fast or not. I know that we have something because we can see the curvature of the membrane, so probably it was inserted in a right way. But I don't know about the velocity, and because normally this phenylalanine could disturb everything. In the beginning, I was thinking, okay, this disturbance could be good because we destroy the organization of the lipids. But at the end, I think that could be hard to put the phenylalanine inserted inside of the lipids. But I never tested before the fluorescence. I think that is a good idea to check the evolution of the entrance of the peptide in the membrane.
Yeah, we could check that doing NMR as well. So yeah, we can have a chat afterwards.
Yeah, for sure. That's nice.
Okay, next question.
Congrats for your presentation. You commented at the end that there could be a lot of mechanism of action, and I believe so. But when you change your peptide sequence, you are focused on the pore formation only or another mechanism? What is the logic of changing the peptides and the flag as well?
Okay, this is a very nice question because in the beginning, I just focus in Q bacteria. So I was not interested in the beginning to see in what is binding or not. And then I was focusing in have a surface of cationic amino acids, and then another one, a very hydrophobic one. So my idea in the beginning, as many others like Bob Hancock and Mike Zaslow, was put in the surface and then make a flip and go inserted of the membrane. But at this point, I believe that was not enough. So the choosing of distribution is okay, is important, but also if I change the position of the amino acids, I can be more effective against certain kinds of lipids. So, for example, if I look to the mammalian lipids and the bacterial lipids, now I can predict what kind of amino acids I can put in a certain place to have more affinity to this kind of one. But now I'm interested because with this paper that we published before, we just get maybe 12 proteins from Klebsiella that appear, and it looks like very important for resistance and for infectivity. So now we are trying to design peptides that bind into these specific proteins and see what happens. So some of them probably not happens nothing, and then the bacteria say, oh, because bacteria is very, very adaptive to even to the peptides. But now the choice is according to the target. In my mind, I don't know if it's the right strategy, but now I'm trying to be more specific. And the tags in the beginning was according to the data bank. So I found a pattern, and then I just follow the pattern that the nature gives to me. But now we are trying to do the same with immune response. If you took the papers of IDRs from Hancock's lab, we have a very nice patterns of immunomodulators that could decrease and improve the inflammation. So we can use these patterns, and also in the information about the interleucin receptors that they touched. So I saw that some of these peptides bind into EL6 receptor, and then we can choose the tags according to the binding to the surface and avoid the interleucin. So now I'm just thinking much more in this direction. I think that I spent 10 years just trying to learn with a lot of mistakes, like this was okay, this was not so okay, but now I'm trying to focus. But this is a good question also.
Okay, any more questions? Okay, thank you so much, Professor Otávio.
Thanks. Eu roubei uma água aqui, eu podia, né? Ah, tá bom. Saber, né? A gente já tá bebendo água. Água alheia, é. Deixa eu botar isso aqui pra cá. Tô aqui, minha filha. Eu não me lembro.
Botando no seu?
Não?
Otávio, no seu?
Acho que é. Isso é um cartão perdido.
Okay, the next talk is Professor Mariana Castro. Mariana Castro is professor for human and comparative physiology of Department of Physiology Science of Institute of Biology from University of Brasilia, Brazil, since 1996. She obtained a doctorate in protein chemistry from the same university. She was a visiting researcher under the supervision of Professor Peter Hopstoff at the University of Southern Denmark during 2011 to 2013. And she is the current president of Brazilian Society of Toxinology, SBTX, from 2023 from 2025 biennium. She has published over 880 articles in Indexed Journal of International Circulation, 7 book chapters, and served a guest editor in the preparation of 3 special issues of Journal of Protein and Peptide Letters. Professor Castro was the coordinator of several grants from Brazilian funding agencies focused on exploitation of Brazilian fauna and research for new drugs and medicines. Professor Mariana, thank you very much.
Thank you, Matheus. It's really hard to talk after Matheus and Otávio, but I will try to do my best. The pointer, let me see if I can work with this. The title of my talk today is Playing with Antimicrobial Peptides, Some Strategies to Improve their Therapeutic Properties. But first, I would like to present myself. I am a member of the Laboratory of Toxinology, and we work with projects to identify and characterize compounds isolated from the animals in general, especially amphibians and venoms from arthropods. And we are also dedicated to the study of the structure-function relationship of peptides, and also to perform the structural characterization of low molecular mass compounds present in venoms and poisons. And our work is developed by myself and in partnership with Professor Osmindo Rodrigues Pires Jr. at the lab. Today, I will talk about the antimicrobial peptides from Anura skin secretions. The class Amphibia is divided in 3 orders with 8,820 species. Anura is the largest one, Caudata the middle one, and Gymnophiona the group with 213 species. Brazil has the greatest biodiversity in Anuras. To register, we have about 1,188 species, and obviously the majority of the species are Anuras. Amphibians are a group that perform the transition between aquatic and terrestrial environments. And to perform this change, several morphological and physiological adaptations were needed. And the integument performed various biological functions, including defense. As we know, the amphibian skin is a rich source of biological active compounds. Here I present the Phyllobates terribilis, that has batrachotoxina, an alkaloid that is used to hunt in the Amazon forest. And we can find in this skin steroids, biogenic amines, alkaloids, and peptides and proteins with several functions: immunomodulatory, hallucinogenic, hypotensive, antimicrobial, insulin releasing, cytotoxic, and neurotoxic. We will talk about the antimicrobial peptides, and these molecules form an ancient defense system against infections. They are forming system present in both plants and animals and microorganisms. And this distribution, ubiquitous distributions, reflects its evolutionary importance. Magainins was the first big hit related to antimicrobial peptides, and it was a serendipitous discovery. By the 80s, Michael Zaslov was working with Xenops levis, and he developed infections. He worked with surgery on these animals, and he was intrigued because those animals didn't develop infections even despite the surgeries were made in non-hysterical environments. So he decided to explore, to identify those compounds, and found 2 novel peptides responsible for those activities. Magainins represents a shield of protection, okay? The key characteristics of antimicrobial peptides, AMPs are typically short, ranging from 10 to 50 amino acids, possess an amphipatic structure with very defined hydrophobic and hydrophilic regions. The biological activity, the antimicrobial effects are normally related to membrane disruption, but as Octavio told us, we have several intracellular effects that we need to explore. And as I told you, yes, we have these targets inside the cell that we need to explore a little bit more. And they exhibit broad spectrum activity. Normally, in general, they are specific for prokaryotic cells because of the organization of the cells with several charged phospholipids, but sometimes we have a broad range of activity, including mammalian cells. About the mechanism of actions, we have the carpet model with some kind of detergent activity, the barrel stave, where the peptides aggregate and form pores, and the toroidal pore, where you have a strong interaction with the phospholipids of the membrane and some kind of distortion of the membrane. Here we have a mechanism proposed for magainin. Magainin form a transient pore, enter the cell and promote the synthesis of REC-A gene and induce apoptosis. Antimicrobial peptides are also able to activate the immune system, and they have, all of them, or most of them, have this kind of dual properties. Our group, our main focus are related to the isolation and characterization of antimicrobial peptides from Brazilian murine species, but we also work with peptide engineer. I will show you two works related to these areas. The first one, the bioprospection of novel antimicrobial peptides. I will present our work with Boana raniceps. This work was developed by the PhD student at the time, Carlos José Correia Santana. And here you can see the chromatographic profile of this skin secretion. Normally, they are very rich, and we focus on this fraction. This fraction was purified by RP-HPLC. The mass was determined, about 3 kilodaltons. And when we performed the chemical sequence of this molecule, we concluded that this fraction was already described from a cDNA bank and correspond to figainin 2. So we decided to characterize this peptide. It has a tendency to form alpha helix and also a very large hydrophobic phase. We performed CD experiments, and the alpha helix was induced by the presence of TFA, and the stability of the peptide was very good. we tested against several bacteria, and the peptide performed very well. We have here gram-negative bacteria, gram-positive bacteria. Yeast, no activity was detected, and we also tested against Trypanosoma cruzi, epimastigotes forms, and the peptide was very potent. We tested against erythrocytes, human erythrocytes, and the activity was low. And against cancer cell, murine melanoma, and breast cancer cells, and again, we have good results. We also tested this peptide with chikungunya virus, dengue virus, and yellow fever virus, but unfortunately, the peptide was toxic for the host, for the mammalian cell. And finally, for this peptide, we evaluated the formation, the activation of human neutrophils by detecting the formation of ROS. And the peptide was able to induce the ROS formation. About the peptide engineering, we have several questions. Why do you need to engineer our antimicrobial peptides? One necessity is related to stability and degradation, because the peptides are susceptible to enzymatic breakdown. About the toxicity and selectivity, because some of them have toxic effects on human cells, the cost of production, resistance development, delivery mechanism, and the immunogenicity. So, and finally, the pharmacokinetics and thermodynamics. We have several strategies to apply to these peptides. So in our case, we decided to invest in the reduction of toxicity and improve selectivity by sequence optimization. In this case, the example that I will show you is oxalatin-4, a weakly cationic antimicrobial peptide with low activity against E. coli and S. aureus, and with a high hemolytic activity. So our goal was to reduce those features by using sequence optimization. We synthesized two sets of analogs. Here I present the data from Fabiano Fagundes Moser da Silva. And we have the first set, we invested in the charge, only the charge. And the second set of analogs, we improved, raised the charge, but we also perform some punctual change to improve not only the division of the hydrophobic and hydrophilic phases, but also to improve the interaction of the peptide with the membrane. We performed the classical serial macrodilution assay. And here we have the results. Here I show you some of the results for pathogenic bacteria, the MIC for gram-positive bacteria, and then we can see that in comparison to oxalatin-4, some of the peptides, the MIC values were improved. We also test again gram-negative bacteria, and we have similar results. Sorry. We have similar results, and you can see here very good results for E. coli and Klebsiella pneumoniae for some of the peptides. We also tested, again, multi-resistant pathogenic bacteria, Staphylococcus aureus methicillin resistance, and again, our cationic, highly cationic peptides performed very well, but we also have a very good one here related to these modifications in comparison to oxalatin 4. We also test against Klebsiella pneumoniae resistance, and again we have a good result with this peptide, and the highly charged ones performed very well. We tested against Candida albicans, and 2 peptides, they gave us good results. We tested against dengue and yellow fever virus. In this case, we have considered dengue virus. We have a very good result with the selectivity index of 59 for this peptide here with several lysine residues. And for yellow fever, only this peptide had a good result, but not so good. In summary, here I show you a panel with the summary of the activities evaluated, and you can see that this peptide performed very well for the tested bacteria, but the highly charged ones were the best, especially this one, considering the activity against dengue virus. Now we are in the we are producing the 2nd generation for this peptide in particular, and also for these 3 here, the highly charged ones, but I don't have the complete results, so I just show you this panel with the 1st generation. I would like to acknowledge the funding research and the support of the pós-graduação em Biologia Molecular e pós-graduação em Biologia Animal for the opportunity to have my students to perform those experiments. And thank you. Sorry for the short presentation, but I was in a hurry because of the meeting, and then I decided to select a few results to give my best here today.
Thank you very much.
Okay. Okay.
Okay, thank you very much for the great presentation, Professor Mariana Castro. It's open to question.
Hi, Mariana. Well, I think that was very nice. It's not a problem to be short. I think that is very nice to be short. Normally I speak too much and the people say, oh, they're sleeping, so short is okay. My question is because you touch in a very, very interesting point. Because normally we are much more worried to find more potent antimicrobial peptides. But you look also to the hemolysis and how to avoid the cytotoxic effects, because this is so important. But normally we don't understand too much about that. We know that when we improve the charges, like you show us, we have more activity against bacteria. But how do you think that we can decrease the hemolytic effect? Because this is also important for drug design
to overcome. Yes. We are working hard on this question because, as you know, when we perform some change, we cannot always predict the results. So we try to improve the charge, but we also try to combine some approach to keep the proportion of the hydrophilic and hydrophobic surfaces. And in my opinion, this equilibrium is the central point to avoid the recognition of mammalian cells in general. Okay? So we'll try to keep this in mind and preserve a limit of the extension of the hydrophobic phase. We usually try to divide this hydrophobic phase with a polar residue, not exactly a polar, but yes, a polar. But let me include this kind of residue, a polar residue, to divide the surface and induce the peptide to avoid the direct interaction with the mammalian cells. This is one of our ideas to avoid this kind of deleterious effect.
I agree with you. I think that the secret will be in the hydrophobic proportion and maybe the distribution, but I'm not sure also. I'm trying many times to change the amino acids, and I never have a consistent result. I never see something that I can control the hemolysis. So I think that this is an amazing question.
Yes, we are focusing on this kind of thing at this moment, but I don't have the final results to
the hammer.
Yes.
Okay, any more question? I have one question. Do you have planned to conjugate your peptide with any molecule to decrease the toxicity and increase the efficacy, or produce formulation, for example?
We are working on pegylation. Yes, this is one possibility, but not with these ones. We have another set of peptides from Ipsiboas albopunctata, and this set of peptides, they are also highly hemolytic, unfortunately, and we are trying to conjugate, to perform the pegylation to avoid this negative effect. Okay?
Okay, and have you tried to test associated with antibiotics in the market?
Yes.
Synergism and addictive effect?
But again, not with those ones. We have performed some tests with peptides from Pithecops species, yes, and also from some analogs, but derived from another group of peptides, the temporins, and for those we have good results in terms of synergisms, synergic effects, but we are still working on these results.
Okay, and about the synthesis of these peptides, you synthesize in collaboration, you pay for them?
Both. We synthesize here in Brasília with Professor Guilherme Grande, but I also pay for some peptides. It depends on the disponibility of Professor Brand to receive our students, and also the disponibility of money to cover those expenses.
Okay, because it's a bit expensive.
Yes, and especially today, nowadays.
It's a limitation for some groups. Any more questions? Okay, thank you very much, Professor Mariana. This session is closed now.
Now I believe you have a coffee break, so we can come back to finish with Professor Fernanda Cardoso.